Thank you so much for that amazing introduction. I can't follow it up with a better talk. I hope I can follow it with a better talk. And for inviting me, for the organizers of ISMB for inviting me to give this keynote. And in fact, this is quite a terrifying honor to open the first post-pandemic ISMB with this keynote. So I decided to go the easiest route possible, not to talk about my research at all, but to show you a whole bunch of movies and videos and pictures of amazing animals. So let's do it. So the Poincaré in 19th century, Poincaré, we're not working, is it? I see. Right, technical issues. Let's do this. Nope, nope. So no. Okay, while we're doing this, I'm clearly going to dance salsa, right? Right? Are we good? We're not good? It's not showing. Okay, now it's showing. We're good. We're good. We're good. Okay. Right. Okay, I'm clearly the test speaker. So, so with that, in 19th century, Poincaré laid out in his beautiful book, Science and Method, what we today consider being the scientific method and the scientific approach. And by the way, if you haven't seen it, beautiful, thin, small book, very concise and explaining everything. At the last page almost of that book, he summarizes the entire kind of his entire manifesto on what is the scientific method, and says that the scientific method consists in observation and experiment, which is what we today consider the standard. If the scientist had an infinity of time at his disposal, it would be sufficient to say to him, look, and look carefully. But since he has not time to look at everything, and above all, to look carefully, and since it is better not to look at all than to look carelessly, which is something I tell all my students all the time, he's forced to make a selection. The first question then is to know how to make this selection. And so scientists have looked for centuries, right? This is how we've been doing science. And in biologists, scientists have looked at nature for centuries and recorded what they saw. And increasingly, with more sophisticated ability to look and to record what they're seeing. And technology helped. It not only enabled us to take more detailed images of what we're seeing, it expanded what we could look at. It expanded the scale of what we could look at, from microscope to the satellite remote sensing. It expanded the scale from the molecules to and below to the planetary scale. And then came genomics. I can't not talk about genomics in this conference, right? So then came genomics, and it changed completely what we could be looking at, what we are looking at. Because we don't have the hardware in our bodies, in the human brains, in the human physiology to look at the genome. And so genomics enabled us to look at that scale, at the genome. And it also, for the first time in the scientific exploration, right, started connecting phenotype to genotype directly. This is through the discovery of the GWAS allows us to connect specific genes to diseases and other phenotypic properties. The comparative genomics, for the first time, really gave us deep insight into the mechanics of evolutionary processes. And we even started changing the phenotype, right? Changing the genotype to achieve the desired phenotype with CRISPR, hopefully ethically. The technology also expanded our ability to look beyond the genome, right? At ecology, at other scales in ecology and wildlife biology. The satellite remote sensing I mentioned, drones and in situ sensors and on-body sensors, as well as the citizen scientists, right, brought in a variety of modalities of looking, of how we can look at nature. And for many of those, in my own research, so this is the part I'm not talking about, the 20 years of research that we've been doing of using all of these modalities, from on-body sensors on baboons and other animals to satellite images, to understand, specifically majority of my research has been focused on how do social animals make decisions, to understand both the individual and the social traits and the interactions between those in decision making in social context. Okay, not talking about that, but please do come to me if you want to hear about that after this talk. The other part that technology brought in is brought us back to our ability to look at scale. It expanded the availability of the recording of what we're looking at in nature, from images to videos in a variety of ways. Images today are the most abundant, readily available source of information about anything, from what you had for lunch and posted on social media to what you see in your backyard or in that amazing safari tour. There are millions and millions and millions of images coming from scientific projects, an intensive collection of data through camera traps and others, to citizen scientists and just tourists going on safaris and whale watching tours and posting on social media, as well as the recent iDigBio project of digitizing biological collections of natural history museums. Millions and millions of images. Just to give you a sense, just the citizen science data from one, the most popular, albeit iNaturalist website, over 40 million, this is 2020 data, over 40 million images of observations of nature, right, currently. It's an incredible source. So can we look at it all? So technology is not only helping us to look at more things, it is helping us to look more carefully. Machine learning and computer vision recent advances, really starting to enable us to not only detect species and organisms and identify their species and be able to track them in videos and images, but also identify down to individuals, even do pose estimations which can lead to behavior identification and tracking, as well as put all of this in the context of environment, because nothing makes sense except in the context of environment and evolution, right? So we can do environmental reconstruction and putting it all together. And so in our work, we have created a platform, Wildbook, based on a lot, this is teamwork and a lot of research and work and engineering, that can take all these millions of images from different sources, any source, whether it's scientific projects, drones, camera traps, citizen scientists, social media, and extract not only species, as in savanna elephant, humpback whale, hawksbill turtle, and gravy zebra, and put a bounding box around each one, so that's detection, right? So that's including that baby elephant hiding behind its mom. But identify individual animals, as in Zippy the zebra, and Joe the giraffe, and Terry the turtle, and Willie the whale.
I wonder, how do they sort through them all and then identify individual animals? Each image is processed through a series of convolutional neural networks and matching algorithms. The first network determines whether there are any animals of interest, in this case, zebras. The next set of networks localize each animal in its own subregion and classify the species. At this point, background segmentation is performed of the subregion to generate a rough mask of the zebra. Once this is done, key points are extracted from the zebra, and feature descriptors are built. These descriptors serve as a sort of fingerprint that can be compared for similarity against previously identified individual animals in a large database. The scores from this comparison allow the system to decide which animals are the most likely matches. When the match has high enough confidence, it's accepted immediately. Matches that have lower confidence are shown to a human expert, who then makes the final decision.
In fact, this is only one of the 7 and about to be 8 different algorithms that we use for individual animal identification. Anything striped, spotted, wrinkled, notched, including the shape of a whale's fluke or the dorsal fin of a dolphin, any biometric feature. And it turns out there are many biometric features that are unique identifiers of individual animals. And together with the information on when and where the image was taken, it can now allow us to track animals, count them, even identify their social network, all non-invasively, right, without having to trap an animal, put a device on it, or taking a DNA sample. This is a page from one of our Wildbooks, Fluke book. This is the Wildbook for cetaceans, and that's Pinchy. She's the most cited individual in this Wildbook. She's a sperm whale, if you couldn't recognize. She was cited more than 600 times. She's a ham, clearly loves it, off of the coast of Dominica. And yeah, you can see also the social network where she's been cited and the map. And so the Fluke book, Wildbook for cetaceans, contains now more than 2 million, in fact, actually just surpassed 3 million images of almost 70,000 uniquely identified individual whales and dolphins, coming from more than 300,000 observations. The BOEM, the Bureau of Ocean and Energy Management, data, all of this whale data is now on this platform, and the newest species in this Wildbook is the killer whale. We'll come back to the killer whale. We even built a bot, an intelligent agent that scours social media and finds all the videos, publicly posted videos of the species of interest, and after analysis and identifying the individual animals in those videos and sending them off to the appropriate Wildbook with an appropriate entry there, you can see the one coming from YouTube in this case. It generates text to post in the comments of the Wildbook saying, hey, of the video, hey, 2 minutes, 46 seconds, we found this whale shark. Here's everything we know about it with a link. And people respond. The most common response we get, wow, this is amazing. How can I help? Because we got people who didn't even think that they could contribute to science and conservation by just posting their vacation video, right, on social media. And suddenly they know something about this animal. They want to know more. They want to contribute to science and conservation. And so the Wildbook for sharks today has more than 17,000 uniquely identified individual sharks. Of those, more than 13,000 are whale sharks. And that data comes from scientists. So this is almost over 90,000 observations that come from more than 9,000 citizen scientists directly contributing data, more than 300 scientific projects, and one very intelligent bot that today contributes more sightings of whale sharks than all the human contributors combined. And 96% of those are unique sightings. So that is significant, right? We're actually, until now, haven't even been using all of those data. Right? So our platform has more than 60 species today, Wildbooks from more than 60 species spanning the entire globe and marine to terrestrial, covering the whole planet. And it's growing. And so why does it matter? What do we get out of it, right? So do all these images give us any additional information? So to get a sense of what we get out of it and what we can get out of it, the International Unit for Conservation of Nature Red List is the official international organization that determines the conservation status of species. So when we say that the species are endangered, it's because IUCN Red List Species Commission for that species determined that the species are endangered. And so out of the 140,000, almost 150,000 species that they track, and they track officially because they're known as the barometer of life, right? So we really defer to them for tracking the status of species. 18,000 almost, their official conservation status is data deficient, meaning we have no idea how many, where they are, what's the species range, is the population, stable, increasing, decreasing. And for 46,000 more, their population status is unknown. And these are not obscure species. These are killer whales. Their official conservation status is data deficient. I cannot go into the field and spend my time putting in policies and actual action to protect the biodiversity of our world. But when I see the conservation status of species as data deficient, as a computer scientist, as a data scientist, I take it personally. This is something we can't do about. And so, the population trend of polar bears is unknown. And even when we do have data for whale sharks, right, the global pre-Wildbook global population size was estimated to be 103,000 with a standard error of between 27,000 and 180,000. Right. I love giving talks to scientists. Right? Right. This is a very scientific way of saying we have no clue. Right. So with Wildbook and with data from images, including images coming from people posting on social media, and a lot of computer vision and machine learning, we got a clue. The most recent global population size of whale sharks was estimated from data from Wildbook. And the conservation status in 2016, this is 2016, they're due for another evaluation. The conservation status of whale sharks was updated from vulnerable to endangered, and the population trend from stable to decreasing, not because the species are doing worse, but because we have the right data to make that determination. And the most comprehensive study on the biology of whale sharks to date was published using data from Wildbook and co-authored by 37 authors, most of whom have not met other than through the pages of Wildbook, because at the bottom of every page, we list all the contributors of data to that specific individual. Because for global species like whale sharks or killer whales, there is not one scientific project, there is not one organization that has the information even about one individual. It is by sharing data and by putting together all of that data that we can really truly understand the migration patterns, the global migration patterns of the species and the seasonality of this migration, that we can start understanding both the evolution and the ecology, right, and the population dynamics. That is the power of images with the technology to be able to see more things and to look at them more carefully, right? The same technology also was used in the first ever full census of the entire species of the endangered Grevy's zebra, or zebra, in Kenya, because 95% of the species are in Kenya. And the first full census was done by ordinary people just driving around the country for 2 days and taking picture of every Grevy's zebra that they saw, preferably from the right side, we asked them. And so 40,000 images later, and covering the full country, we had the information on the population size. And the amazing thing that technology, the ability to identify individual animals from pictures enabled participation of people who've never held a camera before, from school kids and park rangers to tourists with telephoto cameras who did hold a camera before, and US ambassador to Kenya at the time. There are hundreds of people that participated, and our youngest photographer was 3 years old. That's the power of technology that can democratize science, right? And so identifying individuals in all of those 40,000 images, we gave the population size of 2,352 plus minus 72. And Kenya Wildlife Service went complete 180 from saying this is not how we do population size estimates of census to, oh, this is exactly how we're going to do, and now you're on the hook every 2 years. And so we repeated the event in 2018, and more people participated, and Kenya Wildlife Service also asked us to add another species to the count, reticulated giraffe, and this happens to be my absolutely most favorite giraffe in the entire world. We call it Imageomics because it has a heart and a smiley face. Oh, right. This is the appropriate response. Thank you very much. No Photoshop involved. Yes. And so, processing all these images for zebras, we found that the population size increased to 2,800 plus minus 150, and the confidence bounds were so tight that we could absolutely, with pretty good certainty, say that yes, the population size is increasing, definitely increasing. And Kenya Wildlife Service based their entire endangered species policy on this, on these data and on these events. And the event repeated in 2020, and we're gearing for another event in 2023. And the entry for IUCN Red List for Grevy zebras was also updated based on those data. So, are we done? We can do so much with images and with the technology to extract information out of these images. Yeah? No? Okay. But can we really do phenotype to genotype from images? Right. We've been looking at nature. Biologists have been looking at nature and noticing the traits, the behavior, the appearance, the like all these traits that then genomics allowed us to connect to genotype. Right, and if you need a reminder what traits are, right, it's the product of genes and environment and interaction between them. It's everything from the appearance to behavior, like playing dead for an opossum in stress, but everybody remembers the Mendelian peas, right? So the goal, can we, like genomics before that made genes computable, can we make traits computable from images, from observations? And so yes, with that, introducing Imageomics, a whole new field of science that aims to really make traits computable from images, to use images as the source of information about life by using quantitative approaches, to begin with biology-guided machine learning and computer vision, but we're open to suggestions, to extract semantically meaningful biological information directly from images. And it was funded by the National Science Foundation as the Imageomics Institute in October last year. So we're just beginning, so great time to join. And the goal, really, what does it mean to make traits computable? Why is it that, let's say, species classification is not good enough to make traits computable, right? What does it mean? So as an example, here's a project that takes minnows as a whole family of fish that actually are pretty hard. There's about 200, more than 200 of them, are pretty hard for humans to identify different, as classify them into species. So the question we asked, or my colleagues asked, can we directly use computer vision to classify? And the answer is, if you use directly to classify, you can do reasonably well to classify into species. But if you use biological information, such as the phylogeny and taxonomy as structure, as well as the trait ontologies, you actually can not only improve classification into species, they use hierarchical structure of both phylogeny and taxonomy to improve the accuracy of species classification, but you can actually start looking at the salient parts of the image for that classification to start pointing at which parts, biological parts of the specimen, of the species potentially, are important for determining this, for determining the species computationally, right? So there's a long way to go from this to actually extracting biological traits. But what it points to is a couple of things. One is that we may be looking at things, computers may able to help us look at things that humans may not be seeing or not noticing, and helping us to maybe suggest even hypotheses of what should be biologically relevant and salient, right? And there is a lot of evidence that we're not able to look at everything. So this is a recent paper that shows that the polymorphic moth, that humans could not differentiate between species, right? That the color variation that is important for predators, because predators are clearly good at differentiating those species. And the conspecific, so within species, they have no problem identifying their own for mating, right? But humans are not. Computers, on the other hand, machine learning had no problem classifying. So in this case, it's because we're lacking the hardware to see the difference, right? We just can't see in the right range. They all look the same to us. But can computers help us see when humans don't or can't? And so another project that we started asking this question with is looking at mimicry in butterflies. So heliconius butterflies is one of the oldest classic examples of mimicry in nature. So this is the quiz time of the talk. What you see here is 3 sets of column pairs of the 2 clades. So the left column in each set, so the first, third, and fifth, are Heliconius serrato. And that is a butterfly that initially, the species of butterfly, that initially evolved to signal to the predators, hey, I'm poisonous, right? Don't eat me. And the second, fourth, and sixth columns, so right next to it, right, the pairs are Heliconius melpomene. And Heliconius melpomene is mimicking the Arado with half the genome. Right? So it's okay, you've done the hard work. With half the genome and not as poisonous, getting all the advantages, all the benefits of appearing right like the Arado. But so if you look at each pair, right, so the difference is why the first column, let's say, these are all Arado, same species, they're closer related to each other than to its mimic pair, which is another species. The differentiation is because they live in different habitats. That's a whole other story about biogeography and ecology and evolution. But right now, we're interested in the mimicry aspect of it. And so we see, right, we do notice the slight differences between each mimic pairs, but clearly, you know, the false negative for a bird may be too costly. So they're like, oh, close enough, I'm not gonna eat you. You look like that errata thing that I don't wanna eat. But again, conspecifics, melpomenes have no problem identifying melpomenes. So the first question we asked, is there a signal that computers can see, right, in all of this? Can computers identify mimic pairs? Can computers classify the species? Can they point to the parts of the butterfly that are different, that are different between the co-mimics and similar between the co-mimics? So can we really find the parts that are similar and different automatically? And what is similar enough for a bird? Those are the questions that we set out to ask. And we asked the computers to do it. So there's prior work by Jennifer Cattell-Hoyal that showed that you can use deep convolutional neural networks to classify the species. And in particular structure there that they used is the triplet loss, meaning that you give as training, you give triples to the network two that are the same and one is different. And essentially you're asking the network to put everything that you consider the same close enough and everything that you consider different to spread it out far enough. And then you hope that in the end, the same class is clustered together. So we use the same approach, but we explicitly asked about mimics. And so in this asking about mimics, and now I'm worried that you can't actually see it, I'll talk you through it, it's okay. So in specifically asking about mimics and co-mimics, the one thing that we showed, let's see if we can actually see that, yeah. See it? All right, guys on that other side, I'll talk you through it. So one thing that we showed that not only it can classify, so in the top row, the first two are the error on the distance between any two errata, then and Melpomene, that's the first two. So any two errata and Melpomene, the distance in this embedding. Then come the mimic pairs, the distance between mimic pairs. And then come the two between any errata and any Melpomene, sorry, and any Melpomene and any errata. So this is the kind of the non-mimic pairs. And so what you see in the top row here is that there is a signal in different classification regimes. It is putting the mimics closer to each other than it is putting the non-mimics. So great, there is a signal. The question is, what's the signal? The other thing we asked is, if we train it only, let's say, on Melpomene, will it, without ever showing an errata, will it find its mimic? And so the answer in the bottom row, the first one, is a resounding yes. It will find its mimic. So that third box in that graph, that's the distance between errata and its co-mimic pair Melpomene. So it is finding its mimic accurately. Similarly, if you just train it on Melpomene, it will find its mimic pair in errata. So the signal is very strong. The question is then, what is that signal? And so we used transformers, swing transformers, which is a new computer vision technique, to really, literally in this case, transform a Melpomene into errata. That's the first one, or errata and Melpomene. So it's pretty subtle. So that's identifying essentially, what are the parts that need to be changed? And this is identifying it automatically, not just pixel by pixel, but the convolutional neural network layer by convolutional neural network layer. So watch the kind of the twin wing kind of region. And so this is the first kind of evidence that we can, there's a lot of other very, very subtle areas there. So there is the first evidence. And that the interesting part there, that this region corresponds to very specific part of the genome. And so there is some hypothesis that our biologists are getting very, very excited about this, but there's clearly, again, a lot of work to do before we can say things definitively. So I just wanted to show you, because I'm excited, that there is not only a signal, there is actually meaningful signal in this. The other part that we're starting to do is putting these images through the filter of bird, because humans looking at this is not important, right? What we're seeing in this. What's important is what birds are seeing and what butterflies are seeing. So we're putting the images through the filter of bird eyes and butterfly eyes and seeing whether the same part, which is salient, that needs to be kind of changed between the species, how is it different for birds versus butterflies? That's the question of really what's similar and what's different from biological perspective. And so the last story of Imageomics, kind of the beginning of the story of Imageomics, I want to tell you, is back to zebra stripes and leopard spots. So one of the first questions that we ask when we tell kids especially that, hey, we can identify individual zebras from photographs, by their stripes. They're like, oh, a baby zebra stripe similar to its mom's. Great question, right? So the problem is that we have no ability to actually quantify stripe similarity. So if you know, in our brains, we have evolutionary condition prepared to see similarity in faces. In fact, we have a whole infrastructure in our brain to see faces everywhere, right? If you stare at a random pattern for about 3 minutes, you'll start seeing smiley faces, most likely. Depends on the mood, probably. But we see faces because we're wired to see faces, literally. We're not good at comparing stripes. In fact, if I asked you, are the top two zebras more similar to each other than the bottom two? Like, I don't know. We have no, what computer scientists would call middleware to answer that question. We see the stripes, we notice the differences, but we have no software to quantify that difference in our brains. But computers do. That technology that we use for individual animal ID in Wildbook, that has become part of Wildbook, all of that machine learning and computer vision, actually points to the parts of animals that are similar and quantifies the similarity. And so for the first time, we can start answering the question of baby zebra moms or leopards, stripes or spots similar to their moms. And so we're working with scientists, particularly Reuven Yosef, who studies leopards in India. And this is a data set from Indian leopards with information about matrilineal pedigree, not the genetics, actually. And so we can start comparing the pattern of the spots and the similarity in the pattern of the spots to genetic relatedness, or at least to relatedness. And what we're seeing, again, this is just the beginning of the evidence, is, so these are two examples of cubs and moms. So this is a cub and his mom, and this is a cub and his mom. That the faces is where the similarity is most pronounced. It's tantalizing. We're not there yet. I was hoping that that by this talk, I will show you these amazing correlations between like pattern similarity and genetic relatedness. But we're starting to see that signal, and biologists are collecting a data set with microsatellite data, and so the full, both male and female sequences for zebras, for gravy zebras. So we'll go back. So the goal of Imageomics is really to take all the images and our ability to look at nature, augmented by technology, and our ability to record everything that we see, and so augmented by technology, and extract not only, species classification or individual ID, but to be able to say this is this species because it has, an orange belly and black behind the beak. So to go to interpretable AI, to drive interpretable AI to really extract these biological traits from images. And who knows, maybe not only that, but also from videos, and extract behavioral traits and connect them to genotype. So really these biologically meaningful traits, and to understand how social animals make decisions from videos. But ultimately to help scientists look at more things more carefully. And with that, I would like to thank my numerous collaborators and the team of WildMe, the nonprofit, and the founders, the team of the Imageomics Institute, and the numerous collaborators and founders. Thank you very much.
Very visual. Questions? The microphone and your please, if you have any questions, microphone in the middle, and only one that I can see, so please just get in line and feel free to ask. There's another one in there and I can't see. Please.
Thank you. That was a very fascinating talk. How would you put the image of the butterfly through a bird or butterfly's eyes?
Oh.
I was really curious about how you.
Yeah, there are models. Actually, great question. There are models of that translate both the spectrum and the resolution. So there are models of bird eyes, and the sort of computationally we can go from the physiology of the bird eye and translate it to the effect on an image. So both the spectrum and the resolution, and so it changes both in a non-trivial way. So you probably have seen maybe some, like, how do insects or like particularly bees, it's the most common one, see the world, right? So it's shifted spectrum and different and different resolution, not only just the downsampled resolution, but because they have compound eyes, it's like it looks very different in general. So we now can actually do that computationally.
Thank you very much.
Mm-hmm.
Dr. Berger-Wolf, can you hear me?
Yes.
Thank you for the talk. So I was struck by your claim near the beginning that there are biometric features in images that let you identify individual organisms from each other.
Mm-hmm.
And I thought of an example, like think of caterpillars.
Mm-hmm.
Caterpillars and the butterflies they become are the same individual, but you wouldn't be able to tell that from just the images, right? So, and it's an extreme example, but you know, snakes shed their skins and stuff like that, etc. So are there models or principled approaches to integrate info about the developmental trajectory and the physical changes implied therein into this approach of, hey, this is, you know, this caterpillar is this butterfly, if that makes sense?
It's a fantastic question. So, you know, there are species where the pattern is preserved. So we can definitely identify baby zebra as it grows and becomes an adult, and even the distortions of pregnancy or injury, you know, we can, it's still the pattern is determined from birth for zebras and leopards and so on. For turtles, for example, their scale pattern is not determined until a few months, like I think about 3 months. So no, but so it really depends on the species and on the particular biometric feature. But one that project and that question that you're asking is such fun. The reason I'm so excited is because literally this summer we're going to work with a biologist who studies the developmental changes in the pattern on giraffes, right, and how that changes over time. So the pattern is pretty determined, but how it changes and why it changes, and so over time, that is really what we're going to help perhaps do using Imageomics. But the further kind of question of as snakes grow, is the pattern determined, and or how stable and persistent these patterns are for different species, for different features? It's a great question that I think we haven't really been able to ask at scale before, and now can.
Thank you so much.
Inspiring talk as usual. My question is, when you try to show whether a species is endangered, you have to count, and that process strikes me as not easy and non-trivial.
No.
So if you can talk a little bit about counting, especially when you count at multiple points in time. And I can see that you could see the lower bound, right, by identifying ones. But if you have fewer people looking there and the different weather, you know, what are the challenges in counting, and how does that factor into calling a species endangered?
Right. So the determination of whether the species are endangered is done by the Species Commission of IUCN Red List. So they take a lot of information into account. So for that estimate, the global population size estimate for whale sharks was done in 2009 2007 and 2009, they did it twice from genetic diversity of 70 individuals, which is why the uncertainty bounds are so huge, right? So they had a sample of 70 individuals and they did genetic diversity, and so we all know how to do it, right? The founder population, and based on the diversity, they estimated total population, assuming that you're doing random sampling and so on and so forth. So that's why that's one approach how population sizes are done. But specifically for this count using photographs, we went through a lot of actually bias, making sure that the counts are not biased. So this was done over the course of 2 days. So that eliminates a lot of kind of uncertainty in the counting, because and people were spread out all over the country, so they could really cover large areas, and we had a lot of repeated sampling. So because it's over 2 days, so you assume there is no death, birth, in immigration and emigration. You really no, that's fine. Yeah. Oh, it's not stream. No, it's fine. It's fine. It's good. Yeah. Is it showing this? Oh, I see. Okay, let's. Yeah. We do need to do this. Okay. Great. So what it allows us to do is very basic marker capture techniques. And so this chart, the right one that shows the Great Gravy's Rally, it shows that essentially the relationship between the total number of zebras seen versus the number of new animals seen. So essentially by mid-morning of the second day, there were no new animals. And so that's incredible coverage. And we compared the 2016 to 18, and there's really, because you can ask, okay, well, maybe these are only photographs of those zebras that come close enough to humans. You know, you can compare the 2 years and really see that there are not that many zebras unphotographed between the 2 years. And so that gives a lot of confidence of this particular approach, and the reason why other species are now going to be using the same technique. But it doesn't always work, right? And you need to, this is incredible effort to do it repeatedly. This is for one species. So how do we scale these kinds of efforts? And this is where we think that using things like social media posts and citizen science and for some bigger species, aerial photographs, and we're working with data from elephants on using aerial surveys to do more accurate population surveys. And there is, in fact, Department of Interior, and particularly Fish and Wildlife, is starting increasingly to use machine learning and computer vision approaches. And we taught a year ago, a whole team of us taught a workshop to about 300 employees of Fish and Wildlife on machine learning for population surveys. So yes.
Thank you.
Hi, very interesting topic.
I'll start giving shorter answers.
I study population genomics, so this motivated to ask you 2 questions. The first is, what do you think, is it possible to use Imageomics to estimate the migration rate between populations, because you can actually see individuals if you have this time series data? That would be fascinating. It probably is a better estimation than you estimate from genetic data.
So yes, absolutely. And the fact that the biggest one of the biggest outcomes, if we can really compare if there is a correlation, find a way to align the similarity between the pattern, the code pattern, let's say, and the genetics, is that then we can use the code pattern as the proxy for genetics and do things like migration pattern. There's a project where starting conversations, ocelots in Central America and their migration patterns. So they have a good genetic sample of a large population, but not all. And so migration is really, and dispersal is really a big question. So can we use the spot patterns as the proxy? And the big advantage is that this is non-invasive. You can just take a picture and track. And another one is on primates, capuchin monkeys, again, through camera traps, and you can look at the dispersal patterns using facial recognition in this case for primates, which is something we're working on. So the answer is absolutely yes. And the added bonus, if we can really do it for a large number of species, but even for the species that we can, is that we can go back to historical collections and historical photographs, because in many cases we have photographs but not genetics, no biological samples, right? In a lot of different areas. So can we start leveraging these historical photographs to understand the history of dispersals, population dispersal, and other ecological questions.
Right. So you probably can do better than have a constant migration rate, but you can, like, if you have historical data, you can estimate a trend. Maybe you can go even further into the history where you don't have those photos, right?
Exactly. Yes. So we can maybe back propagate. So for the interesting, I don't know if you've noticed, but where is that? Here's come on. Here's the one. For whales, if it works. For whales, once we put up the Wildbook, once Wildbook went live, people started uploading historical data going back to mid-90s, in some cases actually early 90s. So that gave at least some sort of scope of historic data, and we added killer whale to Wildbook, and within months there were thousands and thousands of observations, again going back historically. So by the way, killer whales are going to be data deficient no more by the end of the year, we hope. So yay. That ability to kind of do that from images and then back propagate various parameters is I think a really powerful tool.
That's super cool.
Yeah.
The second question is a bit tricky. Is that how do you can you use this image data combined with like the ability to identify families, the sampling location, to do like unsupervised classification of populations instead of species? Because really don't know, even you have those sampling location, you really don't know which are from the same population.
Yeah, that again depends, I think, very much on the features, the traits, and on the techniques that we use to extract them. Right. So individual ID and that similarity actually allows us, in some cases, to extract some population level traits. But I think even more so, the modern computer vision techniques, particularly that allow us to combined with generative AI, that essentially generates the generic features from a set of features. So we can ask it now to, here's one population, here's another, like generate a set of features that describes and discriminates between those two. And then we can start estimating kind of the distance between two. This is where I see this going. This is where we have a project kind of going in that direction on eggs. So this is collaboration. This is a nascent collaboration with Mark Hauber, who studies eggs, a biologist, and particularly kin selection in eggs. And so mama birds can identify eggs from their nest and reject the ones not from their nest just visually. And Mark Hauber and his group verify that by 3D printing eggs. How cool is that? 3D printing eggs and putting it in their nests And if it's the right pattern, they accept it. So it's clearly not smell or texture, it's just how it what it looks like. And these are eggs with particular pattern, just the spots. There's no color, just the spots. And so, but if you modify these enough, then they will start rejecting. And so what we can start doing now is understand kin selection and then extend it to population level of, because we can generate an egg, not identical looking egg, right? But an egg like that nest, or an egg like that nest, and start interpolate between the two and really delineate the boundaries of kin selection, and then going further to population level traits.
That's a fascinating idea. Thank you.
Yeah.
Amazing and very inspiring talk.
Thank you.
So
Work of many people.
I wanted to know, maybe related to what the previous questions were, do you think Imageomics could be used to inform the tree of life, or would it be different from what it is right now based on just genetic data?
So the question is, can we, could Imageomics be used to reinfer the tree of life? Essentially, can we do phylogeny and taxonomy from images, right? Is that the question?
Yeah.
Okay. Yeah. Can we do phylogeny and taxonomy from images? We've done for many, many, many years. We used to look at, or biologists rather, used to look at specimens and measure all kinds of, for many years, morphological features, morphology, right? And then we derived phylogeny and taxonomy from those, a particular taxonomy. And once we started using the genome as the basis of then we kind of, oops, there were quite a few oops moments there. And I'm putting aside the whole debate of whether species exist as such and what does it mean to have species labels. So, but today, for the most part, we use the genome as the basis for kind of taxonomy and any kind of taxonomic classification and phylogeny. And the big hope and the promise of Imageomics maybe is that we can go back to looking and seeing, and some of it is, these oops moments is because we may be not looking at the right traits. We may not be looking at, not able to see them, like the polymorphic moth, or not paying attention to the right things, like in the minnows, which are, starting to point out that these are parts that we're not looking at. And so in complement, the computer vision and the human vision, maybe we can start looking at the right things, then yeah, let's and leveraging also historical images to do phylogeny and taxonomy from images. Who knows? I'm waiting for Rotten Tomatoes to be thrown at me.
Thank you. Potentially combine the two. I mean, genetics and
I'm not replacing genomics.
Why not?
Just saying, yeah.
So that was an amazing talk, and thank you for the best idea I've heard so far of how to talk to middle schoolers about machine learning. So this will work really well.
Oh, yeah.
I had this broader question, I guess. You said phenotype to genotype. In my world, we talk about genotype to phenotype, right? So this is a very different sort of way of looking at it. So what exactly would you be looking for in terms of sampling, structured sampling, in order to actually be able to map the characteristics you are identifying in the animals that you're studying to actual genotypes?
Great question. It hugely depends on the actual question we're asking about the phenotype, right? And which specific phenotype. And are we talking about the minnows and the species? Are we talking about the individual and and the pattern similarity, or are we talking about the mimicry and why Melpomene can do it with half the genome, right? So I think each one would require a long discussion about what specific aspects of the genome would be relevant for that question. I mean, for butterflies, for example, there's the question about is it half the genome because they essentially figured out, just directly mimicking, or is it, or are they using the same genomes as the Errato for the same traits, for the same to achieve that appearance, or are they just achieving the function and the outcome with through a completely different process? And so that is very much a phenotype to genotype question that will require very careful thinking of how we actually would do that. But I would be happy to have that conversation in whatever context of Imageomics that's interesting.
So just to be a little bit more precise, what I was looking for is if there is some sort of a structured approach where you could say, well, for this particular set of animals, I apologize. Should I repeat?
Sorry, the first question was about sampling approaches but it's the same question. So
well, I'm sort of clarifying, I guess. Is there a structure that you could think of, or maybe something that is in the works, where a particular set of features would define the types of sampling that you would need in order to try to identify the relationship between the phenotype and the genotype?
I don't think we're ready to, beyond the kind of standard sampling approaches where when we're talking about phenotype to genotype or genotype to phenotype, experiments and samples. And I intentionally, you're right, and thank you for noticing, I intentionally put it phenotype to genotype for this talk, right? For a lot of these these questions, there are a few systems where we can really ask these questions, I think, directly. Butterflies is one such system, and these kinds of studies have been done over the years already, so we have some ideas of which genes correspond to, like, the upper orange spots, for example, and so on. And the beyond that, I'm not a biologist. So that's why I'm like, I would love to have that conversation. I'm also would love to bring our biologists into that discussion. But I think that there's one, we can draw on a long history of traits and phenotype to genotype experiments and hypothesis testing. On the other hand, because I think and I hope that Imageomics approaches will point out to new traits that we haven't considered before, then that's where we will have to start looking for some new ideas of how do we connect that to genotype. And just like one of the things that genomics really showed us, that it's not like one gene, one trait kind of thing. It's really the interactions between, quite often, not even multiple genes, it's the interactions among the genes that lead to traits and phenotypes. I think actually in reverse, it's also true. And that it's not, oh, here's one trait and phenotype. It's the interactions among the traits quite often, and these complex traits that we will start popping out of this approach.
This is awesome. Thank you so much.
Thank you.